Today, we're diving into something that sounds like it's straight out of a sci-fi movie, but is actually happening in labs as we speak. We're talking about a totally parallel, invisible form of biology called mirror life. Okay, so just picture this for a second. What if there was a bacterium that your immune system was completely blind to? A virus your cells literally couldn't even see? Or a piece of fruit that would give you absolutely zero nutrition, even if you were starving to death? This isn't just some weird thought experiment, it's the strange reality of what mirror life is all about. So to really get our heads around this, we need to kind of step through the looking glass into a world that's run by a completely parallel biology. And here's the core idea. We're not just talking about one weird organism. We're talking about a potential entire biosphere built from molecules that are the mirror opposite of ours. It would be completely isolated from us biochemically speaking. It would be invisible to our biology, and we'd be invisible to it. They couldn't eat our food, we couldn't eat theirs. It's just a fundamental, total separation. But you're probably asking, how is any of this even possible? Well, the answer is hidden in a really fundamental property of life's building blocks. The concept is called chirality, and you can think of it as life's secret handshake. Chirality. It sounds like a complicated science word, but it's really just a fancy term for handedness. Just look at your own two hands. They're perfect mirror images of each other, right? But you can't place one directly on top of the other and have them line up perfectly. They're non-superimposable. Well, it turns out a ton of the essential molecules that build our bodies have this exact same property. And here's a perfect example, the amino acid alanine. On the left, you've got L-alanine. That's the left-handed version that all life on Earth, including us, uses to build proteins. And on the right, that's its mirror twin, D-alanine. Same atoms, same bonds, it's just flipped. And because of that one simple flip, our bodies almost completely ignore it. This is where that whole hand-in-glove thing really clicks. Think of all the enzymes in your body, those are the little machines doing all the work, as being like millions of tiny, perfectly fitting gloves. And they're all left-handed gloves. So if you try to shove a right-handed molecule into that left-handed glove, it's just not going to fit. The whole interaction fails. And that principle, which scientists call stereospecificity, it basically runs everything in biology. So here's what's so wild. Life didn't just pick a hand at random for every little interaction. Nope, it picked one specific hand for each major type of molecule and just stuck with it across the board. We call this homochirality. As you can see here, all of our proteins are built exclusively from left-handed L amino acids. But at the same time, the genetic code in our DNA and RNA is built on a backbone of right-handed D sugars. And this slide just lays it all out perfectly. On the left, that's us, that's Earth life, built from L amino acids and D sugars. But on the right, you see the hypothetical mirror life, where it's all flipped, D amino acids and L sugars. A mirror organism couldn't use our proteins, and it would find our sugars to be completely useless. It would literally be starving to death in a world that, to us, is full of food. Which, of course, brings up a massive question. Why? Why on earth did life settle on this specific combo of left-handed proteins and right-handed DNA? Was there some deep, necessary reason for it? Or was it basically just a cosmic coin toss? You know, this is one of those questions that keeps origin of life researchers up at night. From a purely chemical point of view, the mirror versions of our molecules are just as stable. They should work just as well. So, was there some hidden force pushing life in this direction? Or did our version just get lucky and win out over the other? All right, so scientists have kicked around a few big ideas. One is the frozen accident theory. Basically, it was pure chance. The first self-replicating thing just happened to use this L and D combo, and it just took over. A more out there idea is a cosmic bias. This theory that meteorites brought a slight surplus of L amino acids to the early Earth, giving our version a little head start. Or maybe a tiny random imbalance was just amplified over and over by chemical reactions. But really, where a lot of scientists are landing is this. Our biology's handedness wasn't destiny. It was a historical accident. It was a contingent event. The dice absolutely could have landed the other way. And because of that, a mirror image biology isn't just a fantasy. It's a completely plausible chemical reality. And that plausibility, well, it's now being put to the ultimate test. We are moving from theory straight into the lab into this incredible and really fast-moving field called synthetic mirror biology.
I mean, just look at this timeline of progress. The speed here is just staggering. In 2016, scientists made the very first mirror image enzyme that could copy mirror DNA. Then in 2019, they made one that could stitch it together. By 2021, they had a mirror enzyme that could amplify big chunks of mirror genes. And by 2022, another one to transcribe mirror RNA. They are literally, piece by incredible piece, building the complete toolkit for mirror life.
And this right here, this is basically the grand finale of all that work. On the left is the central dogma of life as we all know it. DNA gets transcribed into dRNA, which then gets translated into our normal L proteins. But on the right is what scientists have now actually proven is possible. Mirror LDNA can be read by a mirror enzyme to make mirror lRNA, which can then be used to build mirror D proteins. The fundamental logic of life works perfectly in reverse. So this leads us to the ultimate question. We're getting to the point where we can build it, but should we? This technology is opening up a true Pandora's box, and it is full of incredible promise and, well, profound peril. Let's start with the promise, because there are some amazing possibilities here. Since our body's enzymes can't break down mirror molecules, we could make drugs that last way longer and are way more effective. We could store massive amounts of digital information in LDNA, basically a biological hard drive that is completely safe from being eaten by bacteria. And we could engineer mirror microbes for industry that are totally biocontained. If they ever escaped, they just couldn't survive or mix with the natural world. But for every incredible promise, there's a flip side, and it is a terrifying one. A mirror image pathogen, one built from D-Amino acids, would be completely invisible to our immune system, which has spent millions of years learning to recognize only L proteins. It could be a totally unstoppable plague. And what if a mirror microbe, let's say a photosynthetic one, got out into the environment? There would be nothing to eat it. No predators, no parasites, no decomposers. It could just grow without any natural checks and balances and potentially crash entire ecosystems. You know, this quote from one of the researchers really just drives the point home. The potential for a global catastrophe that we can't reverse is not something to be taken lightly. So that's what it all boils down to. By building Mirror Life, we are basically creating a second, totally independent line of biology right here on Earth. It would be a shadow biosphere, biochemically isolated, invisible to our world, and potentially completely unstoppable. And this forces us to face one of the biggest questions of our time. Is this technology humanity's next great tool, the key to unbelievable new medicines and materials? Or could creating a life form that nature has no way to control be our final, irreversible mistake? The science is moving incredibly fast, and the answer is something we're all going to have to figure out very, very carefully.